Hi, everybody. I want to especially welcome all the new faculty. I remember presenting in this conference in 2008, and although I'd been here for a few months at that point, it was really the new faculty members symposium that made me feel part of the bigger community here. So I hope you all had that experience as well. It's a great pleasure to introduce Javier Garcia Bermudez. He was recruited to CRI in 2021. Javier is not a local product. He was born in Spain, and he got his bachelor's and master's degree at the Autonomous University of Madrid, and then a PhD in biochemistry and molecular biology at the Center for Molecular Biology, Severo Ochoa, working with Jose Cuezva on novel regulatory mechanisms in oxidative phosphorylation, centered on complex 5. And from there, Javier went to New York to the Rockefeller Institute, where he did a very productive postdoc with Kivanch Birsoy, using functional genomics, CRISPR screens, basically to identify new metabolic limitations of cancer cell survival and growth, particularly focusing on pathways that allow cells to resist oxidative stress, which is a major bottleneck on tumor growth and progression in vivo. And through those studies, Javier, again and again, was basically able to identify entirely new functions of known metabolic pathways, basically by taking an unbiased approach, has helped us understand the most relevant functions and metabolic pathways that we thought we already understood. And this has become a major theme in the cancer metabolism field. So Javier is a CPRIT scholar in cancer research, and he is also the recipient of a K99R00 award from the NCI. And it's a pleasure to welcome you, Javier.
Thank you. Okay, I think the only problem is with the first slide, so we're good. Yeah, thank you, Ralph, for the introduction, and thank you, really, for organizing this. I'm quite excited about presenting today some of the questions that my lab is asking, the methodology that we're using to answer these questions, and then I will show some unpublished data from what's going to be the first paper from my lab. So in my lab, we're really interested in cellular metabolism, which is this collection of thousands of metabolic reactions. But among these many reactions, we know that oxygen is required for at least 150 of them. So all these oxygen-dependent reactions or processes are very beneficial for cellular fitness and proliferation. But at the same time that cells started to use oxygen for metabolic purposes, they also had to adapt to the negative effects of this molecule, and in particular to the generation of these molecules that we know as reactive oxygen species, or ROS. So when ROS accumulate in the cell, they are deleterious for cell fitness and proliferation. And this is because they can virtually interact and oxidize any macromolecule in the cell. This includes proteins, lipid membranes, or even DNA. So in my lab, we're very interested in understanding this damage of ROS with lipids, which is known as lipid oxidation or lipid peroxidation. And we're very interested on it because of two major reasons. The first one is that when this process is not under control, it leads to a non-apoptotic form of cell death that is known as ferroptosis. And the second reason why we're really interested on it is because there is a lot of evidence that lipid peroxidation is a major limitation of cancer progression. There is now growing evidence that tumors that are exposed to all these stresses have a buildup in lipid peroxides, and that mechanisms in the tumors inhibiting lipid peroxidation increase therapy resistance and metastatic efficiency. And so because of all that, in the lab, we really are interested in studying lipid metabolism in cancer. So perhaps one of the most intriguing discoveries in that field in the last few years is the observation by us and others that some tumors become auxotroph for cholesterol. That means that all these tumors from very different cell of origin, they lose expression in enzymes in the metabolic pathway that synthesizes cholesterol. And because of this, they cannot get their own cholesterol, and they need to take up this essential nutrient from the environment. And as other talks mentioned before, cholesterol is highly hydrophobic, and so it doesn't really travel by itself in the bloodstream, but rather it travels within lipoproteins. So lipoproteins are these large macromolecular complexes that have proteins, phospholipids, cholesterol, and other lipids inside. They can be taken up by any cell in the body, but it looks like these tumors take them up at a higher rate than the corresponding healthy tissues. So a couple of years ago in the lab, we started to ask this question. Do they take it up just to get cholesterol? Or perhaps there is something else within these complexes that tumors like and need to progress? But I mentioned metabolism is a very complicated, a lot of interconnected pathways. So how do we really study metabolic perturbations in tumors? In my lab, to work on this challenge, we use CRISPR-Cas9 genetic screens, in which we take a cancer cell line and we infect it with viral particles that contain a RNA library, usually a metabolism-focused library. We then can subject these cells to different conditions in vitro or in vivo, after which we define gene essentiality. So we decided to use this genetic screen platform to understand the functional consequences of depleting of or supplementing with lipoproteins cancer cells in culture. And to our surprise, when we look at those genes whose essentiality changes the most between these two conditions, we did not find any of the conventional lipid metabolism genes, but rather critical antioxidant genes that belong to the same pathway, the glutathione peroxidase 4 or GPX4 pathway. I didn't mention it earlier, but this system is the major system inhibiting lipid peroxidation and ferroptosis in tumors. So we obviously wanted to validate the screen. We generated full knockouts for GPX4. And it's worth mentioning that these cells in culture do not proliferate. They cannot survive. They just die of ferroptosis in normal media. And they can only survive when we add a well-known inhibitor of ferroptosis. So remarkably, we found that just adding lipoproteins to the media at physiological concentrations was strongly promoting the survival and proliferation of these cells, an effect that was very well conserved across different cell lines and different cancer types. This also made us look at with different eyes to an intriguing observation, which is that in vitro, GPX4 cells die of ferroptosis, but when we take these same cells and we inject them in mice as tumors, these tumors are comparable, almost the same size than control cells. So we started to think about the differences between these two conditions, and it turns out that in vivo, lipoprotein availability is much higher than when we do experiments in cell culture. So we started to think perhaps lipoproteins are having a strong anti-ferroptotic effects in tumors in vivo. To really test this hypothesis, we generated a well-defined genetic model of low lipoprotein availability, which consists of knocking out in the liver of mice ANGPTL3, which is a positive regulator of lipoproteins. So we knocked out ANGPTL3. We also had control mice, and then we injected GPX4 knockout cells as subcutaneous tumors. And remarkably, what we found is that tumors that were implanted in the low lipoprotein genetic model of mice were much smaller and therefore much more sensitive to ferroptosis than tumors that were injected in the control mice. The obvious question next was what component in lipoproteins is really having or driving this antioxidant effect? And let me tell you something, among all the major components that are contained within lipoproteins, many of them, all the ones highlighted here, have actually been reported to have antioxidant properties in the literature. And in our hands, we have validated how addition of each one of these metabolites individually without lipoproteins can rescue ferroptosis. So the bottom line here is that lipoproteins contain multiple antioxidant lipids. And if we really want to induce tumor ferroptosis in vivo, we cannot focus on individual metabolites, but rather we need to inhibit the uptake of lipoproteins by these tumors. So to really identify the mechanisms that tumors employ to take up these antioxidant-rich lipoproteins, we designed a flow cytometry genetic screen platform in which we took a collection of gene knockouts and we incubated them with a fluorescent version of lipoproteins. We then isolated the cell populations that were the most or the least efficient in taking up these lipoproteins, and then we defined the genes that were essential for tumors to take up lipoproteins. So this screen was surprising for two reasons. The first one, we did not find any genes that are the canonical lipoprotein uptake genes. And second, we found a high enrichment on genes that belong to the same pathway, which is highlighted here and is the glycosaminoglycan or GAG biosynthesis pathway. So GAGs are these long linear polysaccharides that are highly sulfated and are usually secreted or on the cell membrane. And in fact, there is evidence that GAGs can interact with lipoproteins, as discovered by here at UT Southwestern by Dr. Browns and Goldstein almost 40 years ago. There is also recent evidence that these glycans could be taking up lipoproteins in hepatocytes and macrophages, but this is the first time we've seen these in tumors. So we wanted to validate these. So what we did was to generate isogenic cell lines of different cancer types that can synthesize GAGs or cannot synthesize GAGs. And indeed, we could see how cells without GAGs could not take up lipoproteins as efficiently, as shown here by flow cytometry. And more importantly, when we injected them as subcutaneous tumor, these tumors were really, really small. They could barely grow. A phenotype that we believe is largely induced by this antioxidant effect, because mice that were supplemented with antioxidants saw this phenotype completely reverted. So what I told you today is that GAGs in tumors are sent to the cell membrane, where they can take up lipoproteins that have a high levels of different antioxidants that inhibit lipid peroxidation and can promote tumor growth. So I want to thank, obviously, the people that did the work, people in my lab, our collaborators in CRI, our funding sources, and thank you for your attention today.
Questions? Okay. Can you wait for microphone, please?
Can you use your lipoprotein to heal atherosclerotic plaque? Do you think it's possible?
Can I use my lipoproteins for
Atherosclerotic plaque
atherosclerosis?
for theranostic?
Yeah, so there is actually some evidence that this pathway, the glycan pathway, is involved in atherosclerosis because macrophages can use them to take up oxidized lipoproteins. So this study was done in tumors because it's the model we use just to study aberrant metabolism. But if we are doing more screens to try to identify the glycans, the specific protoglycans, which are the proteins that have these glycosaminoglycans bound, to know which ones are the most efficient in tumors taking up them, and then try to model them in other disease contexts, which is definitely going to be atherosclerosis.
Okay,
Yes.
thank you.
Okay, we have another question from Siqi. And then we'll go to Ezra, and that's it.
Hi, Javier. I'm just wondering what would be a physiological condition that also uses this pathway in addition to tumor? A physiological condition, for example, yeah.
Yeah, so as I said, it's not the canonical pathway, which could be the LDLR and the SCARB, but there is evidence, again, that in the liver and in the macrophages, there is some of these glycans involved. They're never the major pathway, but they seem to be efficient in some of these cells. So what we think is happening is something very common in tumors, is that they are just hijacking this pathway to overcome the regulation of the other pathways through, we don't know what regulates really this glycan pathway. There might be something upstream of them. So we think they're just taking all the different mechanisms that they can to take up these really antioxidant-rich lipoproteins to overcome the abnormal oxidative stress of that cancers are exposed to. Yeah.
Okay, one last question.
Very interesting. I want to ask you, what lipoprotein did you use in the screen, and does that matter? I would imagine that if you are trying to look at uptake of LDL or HDL or VLDL or other microns, the results might be different. I don't know.
Yeah, that's a great question. Yeah, so we initially, for the fluorescent one, we are using the LDL, but we have seen that this glycan pathway increases the uptake of both HDL and LDL by fluorescent. And we are also seeing very interesting differences in which it looks like HDL has a stronger antioxidant effect in tumors than LDL. So we think there is a lot of room here to really understand which glycans, which pathways, and what are the effects of these two. And we're working to generate these models with high LDL, high HDL, or high of both of them using AV liver in collaboration with Hausu. Yeah, great questions.
Thank you, Javier.